So now let us study about staining techniques. Okay. So why we needed staining? Staining increases the visibility and the contrast of a microorganism, so we can easily see under a visible light microscope. Okay, so it increases visibility and contrast. So this helps in our preliminary identification. Okay, so preliminary identification means nothing whether we can detect whether it is a microbacteria or whether it is a fungi or a protozoa, etc. Also we can differentiate whether it was a gram negative or a positive, and also we can identify special structures like a flagella. Also we can demonstrate motility. So any preliminary identification, it was helpful. So that's why the staining technique is important in lab. Okay, so what are the methods of staining? So there are basically five staining techniques. First one is a simple, then there is negative staining technique, then there is our differential staining technique, then there is impregnation method, then there is special staining techniques. Okay, we will detailedly discuss about all this, don't worry, we will write it down. So first one is our none other than simple staining. So in simple staining, what we do here is that we use common acidic or basic dye. Basic dye is the most commonly used, like our methylene blue carbol fusion. Okay, methylene blue or carbol fusion. So what happens in simple staining is that the entire culture, the entire preparation under the slide will be same color. That is the thing with the simple staining. Okay, so next one is our negative staining. Negative staining technique. So what happens in negative staining technique is that our capsules and our cytoplasm is negative, mostly acidic, so it was have negative charge. Okay, acidic means COO minus terminal will be there, so it is negative inside the capsules and our cytoplasm. So what happens in negative staining is that this stain, the examples of the negative stains are India ink and nigrosin. Both are acidic in nature. India ink or nigrosin and nigrosin, both are acidic, so they are also negative in charge. So it is difficult in order to stain the inside and the capsule, but we will stain entire slide, but we will not stain in the capsules and the cytoplasm. Okay, so that's why it's called negative staining. We will get stain all around the organisms, that is called negative staining. So here you see this is a capsule of Cryptococcus neoformans. So we can see here a halo. This is called a halo. There will formation of there will be a formation of halo. Halo means a hole where there will be no staining. That is called a halo. There will be an appearance of halo. It is the capsule of Cryptococcus neoformans. This is the example for negative staining. Okay. Negative stainings are acidic and capsules are also acidic in nature, so basically they will not stain capsules. And also there are negative staining techniques used in spirochete demonstration. So Congo red is another stain. Congo red is another negative stain used to demonstrate our spirochetes. So these are the examples for negative staining: India ink and nigrosin and our Congo red. India ink and nigrosin generally used for capsules, and then Congo red is used to demonstrate our spirochetes. Then our third method is impregnation method. Okay, so what happens in impregnation method is that You see, some structures are very minute in size, so we cannot demonstrate in a under light microscopy. So we have to thicken it up. So we uses like tannic acid, silver, etc. to thicken up the structures. Okay, so impregnation means we thicken the structure so it will be visible under a microscope. That is the impregnation method. So commonly used is for to demonstrate our flagella and spirochetes. This one is the flagella. Flagella is a small in structure, so in order to demonstrate we have to impregnate it with tannic acid, and then this one is the spirochetes. To demonstrate spirochetes we have to impregnate with silver. Spirochetes. So here our dye used, our stain used is Leveson tannic acid impregnation stain. Leveson, we are impregnating with the tannic acid. Okay, Leveson's tannic acid impregnation stain. So to demonstrate flagella we use Leveson's tannic acid impregnation stain, and to demonstrate spirochetes we use silver impregnation, our Fontana's silver impregnation method. Fontana's silver impregnation. So impregnation method is used to see the structures which are very small in size. Which are difficult to see normally in a microscope. So we in order to see it in under microscope we impregnate it with tannic acid or silver. So this is the impregnation method. Then our fourth method is our special stains, special staining technique. Okay, special staining techniques. So specials means these stains only stain with special structures like our metachromatic granules, etc. Okay, so special staining techniques includes our code. There is a code. It is called ANP stain. Okay, our ANP stain. A stands for Albert stain. N stands for Nice stain, and P stands for Pondeus stain. So all these are used to stain metachromatic granules. You see, metachromatic granules are seen only in less species of bacteria. Okay, so example which is see metachromatic granules are, we can see metachromatic granules in our famous Corynebacterium diphtheria. So in order to demonstrate Corynebacterium we have to use a special stain. Usually we use Albert stain. Okay, Corynebacterium diphtheria, and metachromatic granules are also seen in Corynebacterium cirrhosis, then also seen in Gardnerella vaginalis, and metachromatic granules are also seen in Spirillum. Okay, so for bacteria we use a special stain like Albert, Nices or Pondeus stains. Okay, so you see this is the metachromatic granules. You see a dot like structures here. So these are stained very darkly. So these are the metachromatic granules. This is our normal blood cells. Okay, or systemic cells. You see in Leveson's tannic acid impregnation method we can see here flagella, right? These are flagellas which are impregnated with the tannic acids, and these are spirochetes. Spirochetes are like spring-like structures. You see, these are the spirochetes which are impregnated with the silver, so we can increase the contrast and visibility. Okay, so this is the fourth method, special staining technique. And our fifth method is the differential staining techniques. Okay, this is a special staining techniques. Then our fifth method is differential So differential staining method is used to differentiate whether a bacteria has metachromatic granules or not, whether it is an acid fast positive or negative, whether it is a gram positive or gram negative, etc. Okay, so what are all the differential staining? Let us look. So there is differential stains like our Albert stain. We already talked about Albert stain. Albert stain is a special stain used to demonstrate metachromatic granules. So Albert stain is also used as a differential staining method, because we can differentiate whether the bacteria has metachromatic granules or not. Albert stain. Then is our famous gram staining. Then there is our famous acid fast or Zadan stain. Okay, so these are the differential staining. Now we have to detailedly study about these two, gram staining and acid fast staining. So let us dive into the gram staining. Maybe you probably learn a lot about this in your practicals. So gram staining has four methods, four steps. Okay, so what are all the requirements? We needed a primary stain. A primary stain is our crystal violet. It's a purple in color, purple or violet in color. Okay, this is our primary stain. Then the second requirement is our mordant. Mordant are the substance which is used to settle the primary color inside the cell. So mordant here we use is gram iodine. Then there is our decolorizer. Decolorizer is used to decolorize the primary stain from gram negative bacteria. Decolorizing agent here we use is either acetone or ethyl alcohol. Okay, either acetone or ethyl alcohol, acetone or ethyl alcohol. Here is gram iodine. Then our fourth method is the counter stain. The counter stain is the one stain which will stain the gram negative bacteria. Okay, counter stain. Counter stain is nothing but our safranin, which is a pink in color. Safranin, which is pink in color. Okay, so these are the four requirements in gram staining techniques. So let us dive into how to stain gram. Okay, so first step is our using primary stain. We have to put a inoculum in a slide, and in that culture, in that specimen we have to stain primary color, that is our crystal violet, and after 1 minute, stand it for 1 minute. After that we have to wash it up. Okay, water rinse after 1 minute. Then the second one is the mordant. We add mordant, that is our gram iodine, that is also for 1 minute. Then what happens is that the primary color will settle by the mordant, because there will be formation of iodine crystal violet complex, okay, inside the cell. So it will be difficult for them to move from gram positive bacteria, but from gram negative bacteria they will easily removed. So mordant helps in fixation of the primary stain in gram positive bacteria. Okay, helps in fixation of primary stain in gram positive bacteria. Then after 1 minute, here primary stain is also for 1 minute, mordant also is for 1 minute, then we have to rinse it up. Then we add decolorizer. Our decolorizer is either can be acetone. If acetone we are using, then we have to use it only for 2 to 3 seconds. Okay, if it was ethyl alcohol, then we have to hold it up for 20 to 30. Here 2 to 3 seconds, here ethyl alcohol 20 to 30 seconds. Okay, and after that we have to immediately rinse it out. Just for a few seconds we have to keep it there, and then we have to immediately wash it out. Okay, rinse it out. Then after decolorizing agent adding and rinsing, we use our counter stain, that is our safranin. We can either use safranin or also carbol fusion alcohol also can be used, but most commonly we use safranin. Okay, and hold it up for 30 minutes, and again we rinse it out. So what happens is that the gram positive cocci, you see these are the gram positive cocci which will stain our primary stain, and the gram negative bacilli will stain pink color, that is our safranin dye. These are crystal violet, these are safranin. So these are gram positive, these are gram negative. This is our gram staining techniques. So based on gram staining, there are four principles. There are three principles. There are three principles how a gram staining is done. That means how to differentiate a gram positive and a gram negative. Okay, so there are three principles. First principle is our pH theory. So what is mean by pH theory is in gram positive bacteria, the cytoplasm is more acidic. Okay, cytoplasm is more acidic. So we use crystal violet. Okay, so crystal violet is basic. So what happens is that in gram positive compared with gram negative, it is more acidic. So crystal violet will stain more deeply in gram positive bacteria. That is the pH theory. Then the second theory is cell wall theory. So cell wall theory says that gram positive bacteria has a thick layer of peptidoglycan. About 10 to 100 layers of peptidoglycans are present in gram positive bacteria, whereas in gram negative bacteria peptidoglycan layer is very less. So gram positive have thick peptidoglycan. About 50 to 100 layers are there. So what happens is that this acts as a barrier in decolorizer. So you see, once the primary chain will get inside the cell, it will form a complex with our modern, that is iodine complex will form. So complex has a huge size. So this thick barrier of peptidoglycan act as a barrier in order to move the iodine complex from inside it. So gram positive bacteria will stain the primary chain, whereas when we adding in decolorizing agent in gram negative bacteria, due to less peptidoglycans, this complex can easily washed out. So gram negative will not take the primary chain. Okay, in gram negative, there is thin. So it is more permeable. Sorry about the current. Okay, there is a power cut. So this is more permeable. Okay, so what happens is easily decolorized. Then our third theory in cell theory is concerned with gram staining is lipid content theory. So what happens in lipid content theory is that you see gram positive bacteria has more proteins in the cell wall, because it has more peptidoglycans, more proteins, whereas in gram negative bacteria it has more lipids. Increased lipids are present in gram negative bacteria. Okay, so what happens is that when we add a decolorizing agent, that is alcohol or acetone, okay, when we add a decolorizing agent, there is no dissociation of protein. So pore size will remain the same or will decrease in size and act as a good barrier. But due to the increased lipid content in the gram negative bacteria, when we add a decolorizing agent, that is acetone, when we add acetone or alcohol, what happens is that alcohol dissolve lipid, so pore size will increase. So what happens, the primary stain will easily washed off and it will take a counter stain, that is our softening. Okay, so these are the three theories of gram staining: pH theory, cell wall theory and lipid content theory. The next differential staining technique is our acid fast staining method. Okay, we told there is three differential technique. Our first one is the Albert stain, then there is a gram staining, then there is acid fast staining. So acid fast staining is also important because it is the one of the best method to differentiate mycobacteria. Okay, never forget that mycobacteria are acid fast positive. It is asked entirely throughout. So Zn stain is introduced by our Paul Ehrlich. but later modified the acid fast staining is produced. So it is called Zn stain. Zn staining is modified. Zn. Now it is called as Zn stain, that is Ziehl-Neelsen stain. So they modified this. This is the modified version. Modified acid fast stain. It's a Zn stain, which is founded by Ziehl-Neelsen. Okay. But acid fast staining is introduced by Paul Ehrlich. Later it is modified as Zn stain by Ziehl and Nielsen. So now it is known as Zn stain. So there are three procedures in Zn stain. Okay. The first one is primary staining. So here the primary stain is carbol fusion dye. There it was crystal violet. Here the primary stain is carbol fusion dye. Okay. So along with the primary stain, we have to stain the primary stain, that is carbol fusion dye, for about 5 minutes. Okay. So what we have to do is that after adding carbol fusion, we have to intermittently heat it inside a burner. Not too much heat. We don't want to dry it up, but we have to heat intermittently. So it will fix. Okay. So heat act as a fixating agent. That is a moderate. Here is our heat. Okay. But it is done along the primary staining. Okay. So carbol fusion dye and this heat is done, comes under primary staining methods. And after about 5 minutes of intermittent heating, we have to rinse it out. We have to wash it out, and then we have to do decolorizing agent. Here the decolorizing agent is acid alcohol, that is usually we use sulfuric acid. Okay. So that is the decolorizing agent, and it is done only for about 2 to 4 seconds. Okay. We have to add all our decolorizing agent 2 to 4 seconds. That is, when we add decolorizing agent, immediately we have to rinse it out. Okay. So again we rinse under the tap water. Then our third procedure in Zn staining is our counter stain. Counter stain is our methylene blue. Okay, so what happens is that a counter stain is our methylene blue. So has acid fast positive will stain carbol fusion dye, that is pink in color. So those are the acid fast stain. So here how will the structure will be that methylene blue will color background, and a acid fast thing will color our acid fast organism, that is our microbacteria, spores, sperm heads, etc. Those are acid fast. So those will color as pink in color, and the background will be methylene blue. Okay. So this is the procedure in Zn staining. So let us see what are all the structures which gives acid fast positive. So acid fast positive. Acid fast positive will be like this. Okay, let me draw the slide of acid fast positive. It will look like this under microscopy. There will be acid fast structure, that is our carbon fusion dye, and the background will be methylene blue. This is how acid fast positive organisms under Zn stain will look like. Okay. Now what are all the structures will comes under acid fast positive? The first one is our famous microbacteria. We differentiate microbacteria from acid fast. Okay. So microbacteria. Then there is nocardia. Nocardia is a bacteria. Then there is our sperm head and spores. Okay. Then there is our protozoans, cryptosporidium, cyclospora, isospora. These all are protozoans. So nocardia, mycobacteria, sperm head and spores, and cryptospora, cyclospora, isospora. Cryptosporidium, Cyclospora, Isospora. These are the structures will give us acid fast positive. So that's all about staining techniques. Let us see in the next chapter. Till then, bye.